right, so what's going to happen is I'm going to give you a little bit of time to quickly look through the questions, see what you would answer, and I'm going to go through it. Okay, I'm going to annotate with my thought process, and then we're going to go through things together. Okay, so have a highlighter and a pen and a calculator ready. So can you quickly look at question 1 for me? All right, so question 1 is a multiple choice question on photosynthesis. Okay, and you have to select the one that you agree with on the row that matches the correct description. Okay, so it talks about whether it's an endo or an exothermic reaction. So first of all, you need to remember what endo and exo means. So an endothermic reaction means that energy, okay, is entering that system. Okay, energy is entering that system. Exothermic, energy is going to exit. Okay, so what you need to remember with photosynthesis is that energy is going into the molecules and it is an endothermic reaction. Okay, so it's either going to be A or C. Then you need to recall the equation for photosynthesis to see what's going to be a substrate and what's going to be a product. Okay, so the equation for photosynthesis is carbon dioxide plus water produces glucose and oxygen. All right, and your substrates are going to be before the arrow. Okay, so we're looking at carbon dioxide and water, and your products are afterwards, oxygen and glucose. So the answer for 1 is A. As with all multiple choice questions, get rid of the ones that you know are incorrect, and then if you're stuck between 2 or 3, make best guess. Okay. For question 2, okay, it's about a diagram which shows a potometer. Now, a potometer we use to indicate, measure the rate of transpiration. All right, so if you remember, transpiration is the evaporation of water from the leaves in plants. Okay, so we've got a potometer, you've got your plant chute within the potometer, and you've got what's known as the water reservoir. Okay, water reservoir. All right, now it says what is the structure, why is structure X needed? So with a potometer, what you do is you have an air bubble within your potometer. And as water is taken up by the plant, it's going to draw water along the potometer, and so with it the bubbles. The bubble is going to move towards the plant. So what you are measuring is the distance that that bubble is going to move. Okay, so have a quick think. Why do you think structure X is needed? All right, and why we need water, the water reservoir, is if we open this tap, water is going to go back into your potometer, and that air bubble is going to move back in that direction, and it allows us to repeat the investigation. All right, so the answer is C. Okay, so water . Okay, so it resets the place of the bubble to repeat the investigation. All right, I'm going to move on to question 3. All right, so question 3 and 4 both use the investigation of osmosis using potato. All right, so what we are doing is investigating the rate in which osmosis occurs within plant tissues. All right, so if you can remember, what happens is you have your test tubes, all right, and you have different solutions of different concentrations of sugar or salt. Okay, and in those test tubes, you place your potato or your plant tissue samples. So in the exam, if they bring this up, it's any sort of vegetable or fruit. So they can bring up carrot, aubergine, apple. Okay, the most common one is potato. And you place your samples of your potato. And we use a cork borer to make the samples the same size. Okay, and your solutions have got different concentrations of either salt or sugar, depending on the questions. Okay, so it says the student investigates osmosis. All right, so if you remember your osmosis, that's the rate in which, that's water moving in or out of a plant through a semi-permeable membrane down the water concentration gradient. Okay, so you've got 3 solutions, and at the start we've got the same mass. All right, because we've used a cork borer. To make it constant. Okay, we want to keep the starting mass as constant as possible. And then after 1 hour, your potatoes either lost or gained mass. Now, if we think about the reasons why this is occurring. So if you've got a a plant tissue, and you place it in a solution that's got a higher water concentration, so if it's a higher water concentration, the water is going to move into the plant cell. All right, and that plant is going to gain mass. All right, so it's going to gain some mass. If I place that sample in a concentration of a solution with the same concentration of water, I'm going to have equal movement in and out of my plant cells. Okay, so equal movement. And that means that my plant mass remains the same. So it is not going to change its mass. However, if I place it in a third type of solution where I've got a lower water concentration, water is going to leave my plant cells and go into the surrounding solution. Okay, so I'm losing the water from my plant cells, and therefore my plants are going cells are going to lose mass. And this is key to this practical. If you can understand this and remember this, you'll be able to understand and answer the questions for this practical. Okay, so if water is moving into a plant, it gains mass. If it's leaving, it's losing mass. So for question 3, it says, what is the percentage change? So for this, you need to remember the equation for percentage change. All right. So to work out that, it's the final value minus the starting value divided by the start in value, and we times that by 100. All right, so you're going to get some negative ones and some positive ones. And it says for solution Z. Okay, so we're looking at these values here. All right, and so the answer, if you were to plug it in, so minus 1.7 minus 2 divided by 2 times 100. And if you put that into your calculator, you're going to get minus 15%. So it's B. Okay. So for question 4, it's using the same principle. Okay. But now you get given a graph of the results. So a student investigates osmosis by placing the potato chips and apple into different concentrations of sucrose, so different sugar concentrations. The student calculates the percentage change in length for each chip of the potato and apple, and it has the results. It says estimate the concentration of sucrose inside the cells of the apple. So what you need to focus on is the dotted line. You're going to ignore the other line. All right. Now here, the concentration of the sucrose inside the cells of the apple, okay, you're going to look for where your plant does not change in size, because that indicates that the concentration of the sucrose was equal outside as well as inside. So all you need to do is you look at where it intercepts on that x-axis. Okay, and so have a quick look at the results and think about what one you would do. Okay, and the answer is C. Okay, I'm going to go straight to question 6 next. All right, so if we can turn over the page. Okay, so this is a 6 marker and it's on a practical involving enzymes. Okay, so a student investigates the effect of pH on the activity of enzyme on amylase. So if a question says the effect of something, that's talking about the independent variable. All right, so our independent variable in this investigation is pH. All right, and on the enzyme amylase on starch. So that is a enzyme that we come across in biology quite a lot. Okay, so what it does, it breaks down starch into simple sugars. All right, and it's the amylase. And it's found in our saliva. All right. Now we use an indicator called iodine. So if we add iodine, it turns starch blue-black. All right, so we did this when we did the onion practical. We stained the onion cells and it turned blue-black starch. But when it's simple sugars, so when this starch has been broken down, okay, it's going to remain orange. So we use this as an indicator to see whether the reaction has finished. So at the start of the reaction, it's going to be blue-black because you've got starch. We're going to add amylase, and it's going to break down that starch into simple sugar. So when it's finished, it's going to remain orange. It's not going to change color. Okay. So to change the pH, we use buffers. So as you know from the chemistry, okay, buffers ensures that the pH remains constant. The diagram shows the equipment used in the investigation. All right, so you've got your stopwatch, your wells there as well. So this is the method the student uses. They add a single drop of indicator solution to each of the compartment on the tile. They use one syringe to add amylase solution, then one buffer solution and starch solution to the test tube. They start the stop clock. Every 20 seconds, they transfer some of the solution from the test tube into one of those wells. And they see whether it turns blue-black, which means the reaction hasn't finished, or if it remains orange. And that indicates that the reaction is finished. Okay. They stop the stopwatch when the indicator stays orange. They repeat this whole thing with a different buffer solution. And then here are the results. Okay, so you've got your different pH solutions, and then you've got your indicator when it remains orange. So what this does is it means that reaction has finished. All right, and the longer it takes, the slower the rate of reaction. So here, at 600 seconds, it was still it still changes color, which means the reaction hasn't happened. So it indicates a very slow rate of reaction. Okay, so at pH 3 and pH 11, we had slow rates of reaction because it was still changing color after 600 seconds. At 60 seconds, so at pH 7, it only took 60 seconds to change color. And that is the quickest rate of reaction. All right, so if I was to sketch a graph, so if I was answering this question, all right, they just give you a table. It's quite hard to picture things in a table, so I would sketch it. So I'd put my pH there, and then your rate quickly there. All right, and then you've got 7, 5, 3, 9, and 11. So at 7, you had the quickest rate. Okay, and then at 5, 7, and then at 11 and 3, you did not have a reaction. So that's what my graph would look like. Okay, because my rate of reaction was quickest at 7. Because the question then goes on to say, on the next slide, explain the pattern of the results. Include ideas about the mechanism of enzymes in your answer. Okay, so you need to explain why there was a quicker rate of reaction at pH 7 and no rate of reaction at 3 and 11. All right, so bear with me while I right, so when it says explain, you have to give reasons for. All right, make sure that you give a reason for. All right, make sure to include ideas about mechanisms of enzymes in your answer. So whenever it says about mechanisms of enzyme action, you need to remember the fact that you've got an enzyme. In that enzyme, you've got an active site. Now that active site is where the substrate binds. All right, and what you need to remember about your active site is that your substrate, which is the molecule that binds to that enzyme and either gets broken down or built up, okay, is complementary in shape. So your substrate is complementary shape, so it's specific to that. All right, so whenever it says about mechanism of enzymes, make sure you bring in this knowledge. Okay, so your substrate is going to bind to your active site in your enzyme, get broken down into its products. And it needs to talk about the pattern of the results. Okay, so you're going to bring in this graph here. Okay, so your pH 7, okay, and your pH 3 and 11 had no rate of reaction. So first thing that I would do, all right, is I would comment about the fact that pH 7 has got the fastest rate of reaction. Okay. So rate of reaction is fastest at pH 7. Now, the term that we need to use when it talks about how when it's the fastest rate of reaction with enzymes, is we talk about that as being its optimum. Okay, so this indicates that pH 7 is the optimum, so where it works at its most quickest, pH for amylase. Okay, so I've talked about where it happens at the quickest. Okay, I'm then going to also bring in the fact that the rate of reaction slowed on either side of pH 7. Notice I've got bullet points because I'm spacing out my answer into separate little bits. Okay, now with temperature and pH, if the temperature is too high or the pH is below or above the optimum, the enzyme is denaturing. So the shape of my enzyme is changing and my active site shape is changing, which means my substrate is no longer going to be able to bind to my active site. All right, so I've commented on my result and I'm going to now explain it. So I'm going to say because the enzyme is becoming denatured. And then I'm going to explain what that is. So therefore, the shape of the active site changes, and starch, our substrate, can no longer bind. And all that's left to do is comment about the pH of 3 and 11. So at pH 3 and 11, the enzymes are completely denatured. And starch has no longer been broken down. Now, as you can see, I haven't written much, but that would have got me 6 marks. Okay, so it's not always about the amount of content, it's just the quality of the content.